Welcome. This presentation examines one of the most studied genes in human biology, TP53, and its central role in genomic stability, tumor suppression, and cancer evolution. The research and analysis presented here are developed and delivered by Brian Geniuson, research scientist. This lecture proceeds systematically from molecular structure to mutation architecture, cellular dysfunction, systems-level disease mapping, and translational therapeutic strategy. This is an educational scientific discussion intended for informational purposes only. TP53 encodes the protein p53, a transcription factor widely recognized as the guardian of the genome. Under normal physiological conditions, p53 is tightly regulated and rapidly degraded through interaction with MDM2. When cells experience stress, including DNA damage, oncogene activation, hypoxia, or telomere shortening, p53 stabilizes and activates transcriptional programs that regulate cell cycle arrest, DNA repair, apoptosis, senescence, metabolic control, and therapeutic signaling. Through these mechanisms, p53 maintains genomic integrity. Without functional p53, cellular stability progressively declines. TP53 is the most frequently mutated gene in human cancer, with alterations observed in approximately half of solid tumors. Most mutations occur within the DNA binding domain. Common mutation classes include missense substitutions, nonsense mutations, frame-shift insertions or deletions, dominant negative variants, and gain-of-function oncogenic mutants. Hotspot residues such as R175, R248, R273, and R282 are particularly vulnerable because they influence structural integrity and DNA interaction. These mutations may abolish tumor suppressor function, interfere with residual wild-type protein, or confer novel oncogenic signaling properties. Loss of functional p53 disrupts multiple regulatory checkpoints. Without adequate p53 activity, p21 is not induced, cell cycle arrest fails, DNA repair pathways are insufficiently activated, and apoptotic mediators such as BAX and PUMA are not transcribed. This permits survival of genetically damaged cells and accumulation of additional mutations. Certain gain-of-function mutants may further promote invasion, metastasis, inflammatory signaling, chromatin remodeling alterations, and metabolic reprogramming. At the cellular level, loss of p53 contributes to genomic instability, chromosomal rearrangements, elevated mutation burden, dysregulated mitochondrial signaling, and metabolic shifts toward aerobic glycolysis. These alterations increase clonal heterogeneity within tumors, enhancing adaptability and therapeutic resistance. TP53 mutations are prevalent across multiple organ systems, including breast carcinoma, lung carcinoma, colorectal carcinoma, ovarian carcinoma, pancreatic adenocarcinoma, glioblastoma, sarcomas, and hematologic malignancies. Germline mutations in TP53 result in Li-Fraumeni syndrome, characterized by early-onset multi-organ cancer susceptibility. At a systems level, TP53 dysfunction destabilizes tissue architecture across proliferative domains of the body. Therapeutic strategy depends on mutation context. If TP53 remains wild-type but suppressed, MDM2 inhibitors may restore function. For specific missense mutants, reactivation compounds such as APR-246 have been investigated. Additional strategies include synthetic lethality approaches targeting ATR or WEE1, DNA damage pathway exploitation, immunotherapy stratification, experimental gene therapy, and CRISPR-based correction research. Precision oncology begins with accurate mutation classification. Two-tier evidence supports this framework. Tier A evidence includes large-scale genomic sequencing datasets from international cancer genome consortia demonstrating high TP53 mutation prevalence across tumor types. Tier B evidence includes extensive clinical oncology research correlating TP53 mutation status with tumor aggressiveness, prognosis, and treatment response. Mechanistic validation across cellular and animal models further reinforces the biological foundation. Within a systems biology framework, TP53 interfaces with genomic instability, cellular senescence, metabolic rewiring, immune surveillance, and aging-associated pathways. It represents a central regulatory node linking mutation to organism-level pathology. TP53 provides a foundational example of how a single genetic alteration can propagate from molecular disruption to cellular dysfunction, tissue instability, and systemic disease. Understanding this cascade enhances precision medicine, mechanism-based therapeutic design, translational oncology strategy, and genomic disease modeling. Full scientific literature references are available in the video description. For continued structured discussions in molecular and systems biology, explore the MUT-PATHO-MASTER series playlist. This presentation is provided for educational and scientific discussion purposes only. It is not intended as medical advice, diagnosis, or treatment guidance. Clinical decisions should be made in consultation with qualified healthcare professionals.